Hello everyone and warm welcome to the pre-recorded training series for developmental neurotoxicity in vitro battery. The series are hosted by the OECD Environment, Health and Safety Division of the Environment Directorate. My name is Magda Sachana and I am a policy analyst to the test guidelines and hazard assessment programs. Before we go further, I would like to let you know that although the presentations are pre-recorded, we do value your feedback. So if you have any questions or remarks, please don't hesitate to contact me in this email address. So what was the motivation behind this training series? The biggest motivation was the publication of the initial recommendations on evaluation of data from the developmental neurotoxicity in vitro testing battery. It is worth mentioning that the publication of this guidance was an international effort and many more experts than the ones that you'll see in the recordings contributed with the drafting, reviewing, or development of case studies. So here you have the QR code, so I will encourage you to download the document and read it in advance or at least in parallel with this training series. So what is the scope of the training series? The last 2 years since the publication of the guidance, we've heard that the regulatory uptake of the DNT-IVB requires training in evaluation of such data. So these training series were developed with the aim to provide an understanding of the DNT in vitro battery and educate participants on the methodologies, regulatory applications, and interpretations of DNT-IVB data. That's why we built the training around 4 sessions. Session 1 provides a broad overview, introduces key concepts and methodologies associated with DNT-IVB. Session 2 delves into case studies, really highlights the regulatory application of DNT-IVB data across different OECD member countries and industries. Moving to session 3, this session explores the interpretation and application of DNT-IVB data for chemical assessments. It focuses on quantitative in vitro to in vivo extrapolation and physiologically based kinetic models that are used to predict fetal and infant exposure. Finally, session 4 provides practical insights on accessing and analyzing DNT-IVB data. It really offers hands-on training and comparison of different pipelines where data is stored. So for who is this training? The training is tailored for a diverse audience, including regulatory professionals from governmental agencies involved in chemical safety evaluation, industry experts involved in chemical testing and safety assessments, researchers and scientists in developmental neurotoxicity, all the people who develop the methodology, as well as academic professionals and students with an interest in DNT. So if you are looking to deepen your understanding in the use of DNT-IVB data for regulatory applications, this training is for you. Thank you very much for watching, and please continue watching the rest of the training. If you have any questions, here is again my email address, and may I take also the opportunity to thank all the presenters for putting together the slides and the recordings. So thank you very much.
Hello and welcome to today's webinar, the DNT Developmental Neurotoxicity in Vitro Battery IVB, Assays and Underlying Biology. My name is Fritsche. I'm the director of the Swiss Center for Applied Human Toxicology, the SCAHT, in Basel, Switzerland. I am one of the DNT in vitro battery developers. I love to give presentations, and I would like you to understand the nature of the DNT in vitro battery. The overarching concept of the DNT-IVB is mimicking neurodevelopment in vitro by assessing individual cellular key neurodevelopmental processes in a dish. Therefore, this is not one individual assay that concerns the battery, but it's a battery of different tests comprising the DNT-IVB. Before I come to the details of the battery and also the assays, I would like to briefly glimpse into the underlying biology of brain development. I promise this will be brief and only necessary for understanding the assays. During brain development, key neurodevelopmental processes shape the brain over time. During embryogenesis, to be seen on the left here, the neural tube is formed and segmented, followed by further development and growth in the fetal period with increasing complexity pre and even further postnatally. On the bottom, you can see the temporal resolution of these processes like neuroangliogenesis, migration, neurite outgrowth, here shown as neuritogenesis, and myelination are shown. In general, growth, differentiation, and networking characterize this very complex and orchestrated process. In humans, brain development is actually finished not before reaching 25 years of age. Especially during the fetal period, the cortex is formed. This is a period of very active development where the foundation is laid for the future outcome of neurodevelopment, the functioning brain. Here, the proliferation of stem cells, which is followed by migration of radial glia cells, which also belong to the group of neural stem cells, and further differentiation into neurons, astro, and oligodendrocytes are key. So these cells, they have to migrate and have cell-cell interaction that are needed to form connections in between the cells, the so-called synapses, resulting in functional neural circuits and networks. And so the cortex does play a crucial role in higher order functions of the brain, which are sensory function, motor functions, cognition, also language, emotions and social behavior, learning and memory, as well as consciousness and self-awareness. So these are all functions that are transported by the cortex. Based on this basic knowledge, the key neurodevelopmental processes, which are schematically depicted on the right, can be summarized as follows. So brain precursor cells, also called neuroprogenitor cells or neuroprecursor cells, have to proliferate because the brain has to grow in the end. But the superfluous cells, they also undergo apoptosis, especially after they have been differentiated into neurons, because you want to shape a brain that consists of cells that are absolutely necessary for producing the functions. So these cells also have to migrate and they have to differentiate, so they have to take place in a coordinated manner. Neurons have to grow, expand neurites, synapses have to form between the neurons as part of the establishment of neural networks and axons. They need myelination to perform properly and have the saltatory conduction taking place. Moreover, microglial cells, the immune cells of the nervous system, which migrate early into the developing brain from the periphery of the embryo, also participate in many of these processes, and a lot of functions of these microglial cells are not completely understood yet. So the relevance, and here especially the human relevance of these key neurodevelopmental processes, becomes obvious when the homeostasis of these processes is disturbed. So this is actually observed in a variety of human neurodevelopmental diseases. For example, syndromes with hyperactivity, like, for example, the autism spectrum disorder, produces excess neurons, while diseases characterized by depression and anxiety can see a reduction in neurons or neuronal subpopulations with decreased connectivity. So all these are very nicely summarized here in this article by Griffin et al. In 2022. Now, I gave you a very brief overview over basic principles of brain development and the relevance of key neurodevelopmental processes, also with regards to neurodevelopmental diseases of humans. And next, I will provide you now with an overview over the DNT in vitro battery. The DNT-IVB is a battery of human-relevant in vitro assays, and they were designed to assess the potential of substances to interfere with brain development. It focuses on key neurodevelopmental processes based on human biology, like I explained to you earlier. On this basis, 17 test methods were set up covering 8 key neurodevelopmental processes as endpoints with 8 test systems from 3 developer labs, the lab from J Shafer at the US EPA, Marcel Leist from the University of Constance, and my former lab in Düsseldorf, now Katharina Koch's lab at the IOF in Düsseldorf in Germany. The round icons here indicate these 8 different endpoints, and the next slide now will summarize the 17 different test methods that in the end form the battery. Here, each of the frames now depicts one of the F8 endpoints, indicated again by these round icons. The symbols on the right inform on the developer's lab, like I said, the IOF, the EPA, and the University of Constance, and in the middle you find the names of the individual test methods. H is referring to human and R to rat-based test methods. So this battery was assembled many years ago according to the test methods readiness, which can be found in this article from Balprice et al. In 2018. Now I will walk you through the 17 assays and respective data generation with the goal of familiarizing you with the individual tests. So the prerequisite for every assay is a test system, which is a reproducible cell culture and the challenging of the respective cells. So based on this test system, the test method is developed here. The test method is developed, so exemplifying here this very valued publication explaining this process, how to get from a test system to the test method by Crofton et al. From 2011. So once the test method is set up and the testing scheme is established, compounds can be tested, and then concentration response data can be analyzed and the compounds can be classified, and I'll explain this a little bit more in detail later. I will give you now an insight into one of the in vitro methods from the DNT in vitro battery, which is the human neurosphere assay that my lab developed over the last 2 decades when I was still in Düsseldorf. So these are human neurospheres, and they are primary human fetal progenitor cells from human brains. So neurospheres make use of the fact they consist of neural stem or progenitor cells, as you can see on the left here, when they are proliferating as so-called spheres. And these cells can become brain effector cells, neurons, radioglia cells, astrocytes, and oligodendrocytes upon plating, which you see on the right here. So when you stain for neurons and oligodendrocytes, then you get, and the nuclei in blue, you get these images that you can see here on the right. So this neurosphere assay covers 7 of the 17 DNT in vitro battery assays and is capable of addressing 5 of the 8 DNT endpoints of the in vitro battery. Specifically, the test methods NPC 1 to 5 assays cover, like here, NPC 1, the neuroprogenitor cell proliferation, which can be measured directly by BRDU incorporation into the floating 3D neurospheres. Moreover, upon plating, like I said, so the cells migrate radially out of the sphere, and you can see a time-lapse microscopy here on the bottom, where you can easily appreciate that the cells have different shapes and they actually form connections that they form and loosen again. And so this is a very active and interactive procedure. And this radial glia migration is the NPC2 assay. In the NPC 3, 4, and 5 assays, neuronal differentiation, oligodendrocyte differentiation, but also neurite outgrowth is assessed, and the migration of the neurons and oligodendrocytes, they also belong to the NPC 2 assays. And so NPC 2 to 5 are multiplexed so that in the end the neurospheres are only comprised of 2 different actual assays, but with 2 to 5 being multiplexed. Also, astrocytes belong to the cells of the neurosphere assays, but these assays concerning astrocytes are currently not in the battery, but belong to the future development. But effectively, within the migration area of the neurosphere, you have all the different neural effector cells, all but the microglia present. Besides the assays developed by my former lab, the University of Constance established test methods for migration of neural crest cells, like shown here on the left, but also effect on neurite outgrowth of central nervous system and peripheral nervous system neurons. Here the different test systems are applied. So the neural crest cell migration the basis are iPS-derived neural crest cells, while the effect on neurite outgrowth of the central nervous system neurons are immortalized dopaminergic midbrain precursor cells, so-called LUMA cells, while the peripheral nervous system neurite outgrowth assay, which is UKN5, are based on iPS-derived peripheral neurons. So all these are neuronal or neural crest cell monocultures without the presence of glial cells. Moreover, the EPA lab developed assays for multiplex proliferation apoptosis assays, here seen on the left, which are using HNP1 cells, which are embryonic stem cell derived neural progenitor cells. Also neurite outgrowth assays with iPS derived neuronal monocultures or rat mixed culture test systems assess neurite outgrowth. And then the more advanced endpoints of synaptogenesis and neural network formation are covered by rat primary cortical cultures. So with all those assays from the 3 developers labs, the large variety of key neurodevelopmental processes is covered within the DNT in vitro battery. Each assay that I have shown you now is accompanied by cytotoxicity and or cell viability assays to distinguish a specific effect on a key neurodevelopmental process from a cytotoxic effect. And this is important when it comes to compound classification. But getting there, so now how is the data produced with these assays processed? Individual data points of concentration response data is summarized and curves are fitted. For each assay, a specific benchmark response level is defined that is based on the assay's baseline variabilities. The output is the respective benchmark concentration at the benchmark response level. To make the influence of the biostatistics on assay outcome transparent, we actually published these 2 manuscripts that are depicted here on the right on pipeline comparison, for example, between the ToxCast TCPL and the IUF CR stats, and also the ICE pipelines, showing the possible influence of biostatistical evaluation for compound classification. Classification into these 4 categories is performed by assay-specific prediction models, here exemplified for the IOF assays. But details on all the assays of the battery and their classification model and their prediction model can be found in the OECD DNT Initial Recommendations number 377. So specifically here we distinguish if a compound produces no hit, that means the specific endpoint, and also the cytotoxicity or viability is not altered at all. Moreover, we talk about a specific hit when the effect on the specific endpoint and the cytotoxicity or viability can be distinguished, which means that the confidence intervals at the benchmark response level of the specific endpoint and the viability cytotox endpoint, they do not overlap. If they overlap, that can be seen on the lower left here, we talk about an unspecific hit because we cannot really distinguish between effects on the general viability of the cell and the specific effect on the key neurodevelopmental process. And then we have borderline hits where either we have not tested high enough, or also the confidence interval doesn't really cover the benchmark response level. So now that we know how to analyze the individual assays, it's also important to understand that not the individual assays, but the outcome of the whole battery will inform on the DNT potential of the substance tested by the DNT-IVB. For example, looking at the 17 assays that are depicted here now by these different circles, when only one endpoint is affected, here like an example for assay 17, or you might even have multiple endpoints affected, maybe at much higher benchmark concentrations, or multiple with a similar benchmark concentration, we are looking at the most sensitive endpoint. So in this case, in this example, this would be the effect on assay 16, which would then be the regulatory relevant endpoint. And this might as well be multiple endpoints, and this will be shown in later webinars in this series. So staying with my example, assay 16 being affected, and this endpoint here is oligodendrocyte differentiation. Now we want to link the effects of the battery to the adverse outcome pathway because we want to substantiate the relevance of the key events of the battery. So in this example of the oligodendrocyte differentiation, there is a small AOP network already available in the AOP wiki. This is AOP 525 that converges a variety of molecular initiating events here to the endpoint oligodendrocyte differentiation. So looking at the network view of the AOP wiki here, so this AOP is linked to other AOPs that are depicted here by the gray circles, mainly through the adverse outcome, which is impairment of learning and memory. So for the use of the battery, addition of more AOPs for all necessary key neurodevelopmental processes or endpoints of the battery is actually strived for. So momentarily, there are still large gaps for DNT AOPs in the wiki, but these are being constantly filled. And this brings me now to the ongoing activities. So what is happening in the field of the DNT-IVB? So on the one hand side, there is more compound testing going on. And one example is the very large project of the US Division of Translational Toxicology, the former NTP at the National Institute for Environmental Health Sciences. Then there is testing going on within the Horizon Europe Project Park. Then in the DNT-RAP1 project, which is sponsored by EFSA, the IOF, the EPA, and the University of Constance, and also the NTOX are actually testing pesticides in the DNT-IVB. Then we have assay refinement or further development going on, and again, within the Horizon Europe Park project, but also the Chiasma project, assays are refined and assessed. Then the Brain Health Project, also sponsored by EFSA, is a project that has the focus on transcriptomics related to glia-related endpoints, which are at the moment a little bit underrepresented in the DNT-IVB. And also further assay development is going on at the EPA and also at the Johns Hopkins University under SCAHT at the University of Constance, the Norwegian Institute of Public Health, the IOF, and the University of Lausanne in Switzerland. Moreover, case studies, which are generally performed through AOP or IATA projects, so integrated approaches to testing and assessment, take place within also the PARC project, but also the ASBIS cluster with the two major projects, OnTox and Risk Hunter, which both work on DNT, and assays of the IVB are producing case studies. And also the EFSA-sponsored DNT-REP4 project is developing IATAs in combination with the DNT-IVB assays. And last but not least, the AOP hub from the SCAHT, so from my institution, is constantly now sponsoring development of DNT AOPs. Another very important current activity for the regulatory application of the DNT-IVB is the lab-to-lab transfer, which is currently ongoing. So from the 3 different test method developers labs, the EPA, the IOF, and the University of Constance, all battery assays are transferred to the contract research organization DNTox, which is a startup company out of the IOF. And this serves to show that the assays are transferable, which is a prerequisite for also the validation of the assays. But at the same time, the assays will also be available, which is important for the regulatory application. And with this, I'm coming to the end of this webinar, and there are many people have been and are involved in the DNT in vitro battery. For one, these are all the test method developers labs, all the students, all the postdocs who have been putting a tremendous amount of work into the development of the assays and into the scientific validation. But I would like to say that without the support of the OECD by the DNT in vitro battery ongoing expert group, and especially EFSA and the US EPA supporting especially the academic researchers almost from the beginning of the development of the battery, we would not be standing where we are standing. Also, the constant support by the Joint Research Center of the European Commission, but also by the Ministry of the Environment and Food of Denmark. And last but not least, also the founders of the DNTox company, which have been embarking on this journey of startup to make the assays available. Without them, we wouldn't be where we are. And last but not least, thank you very much for your interest in the battery. And if there are any questions remaining, please feel free to contact me or anybody else who is now contributing to this webinar series. Thank you very much. Have a great day.
Welcome to the module on establishing confidence in the use of data from the DNT in vitro battery. My name is J Shafer, and at the time of this recording, I'm a senior research toxicologist with the U.S. Environmental Protection Agency. Because I'm an employee of the U.S. Environmental Protection Agency at the time of this recording, I need to present this disclaimer. It states that this presentation has been reviewed by the EPA and has been approved. It does note that any mention of products is not a recommendation for use or an endorsement, nor does anything presented in this presentation represent the policy of the U.S. Environmental Protection Agency. These views are mine and mine alone. What I'd like to accomplish in this presentation is to give you a background that establishes confidence in the use of data from the DNT in vitro battery for regulatory decisions. I want to discuss the biological relevance and relationship of to current regulatory endpoints of the DNT in vitro battery, discuss how the DNT in vitro battery meets current frameworks for confidence, and provide some brief updates on assay transfers and other future directions for the DNT in vitro battery. The key premise underlying the in vitro battery is that if we evaluate the effects of chemicals on key neurodevelopmental processes that are required in order for the nervous system to develop, such as proliferation, migration, differentiation, apoptosis, and so on, that chemicals that perturb these functions or these key neurodevelopmental processes have the potential to cause developmental neurotoxicity. These key neurodevelopmental processes can be shown to be related to different environmental agents that are known to cause developmental neurotoxicity, as well as different clinical conditions that are known neurodevelopmental diseases. And so on the far left-hand side in this table, we have key neurodevelopmental processes. In the middle column are different known neurodevelopmental chemicals or physical damage that cause changes in this key neurodevelopmental process in vivo or in vitro. And finally, on the far right-hand side here, we have different neurodevelopmental diseases where changes in the key neurodevelopmental process are part of the sequelae of the disease, or at least common in the disease. And so what I want to be clear about is I am not saying that the chemicals in the middle column cause these diseases. That is not the case, or that is not what I am asserting here. But that both the chemicals in the middle column cause changes in the key neurodevelopmental process, and that changes in the key neurodevelopmental process are part of the neurodevelopmental diseases in the far right-hand column. Furthermore, these changes in these key neurodevelopmental processes can be mapped to different in vivo outcomes that are commonly assessed in the in vivo guideline studies. So for example, changes in proliferation, apoptosis, differentiation, and migration can give rise to various different measures of brain malformation and brain size that contribute to changes in gross morphology. Cytotoxicity, apoptosis, necrosis, and so on can give rise to changes in the histopathology that are often measured in the in vivo guideline study, as well as morphometry. And finally, changes in synaptogenesis, network formation, and others can give rise to changes in learning and memory and motor activity. And so these key neurodevelopmental processes contribute to changes in the gross morphology, histopathology, and morphometry and learning and behavior that are measured in the in vivo guideline study. I'd like to spend the bulk of the presentation today talking about how the different DNT in vitro assays map to the criterion that have been established for establishing confidence in the use of data from NAMs for regulatory purposes. And so these are two different constructs that are, as you can see, very similar, that were published by van der Zalm in 2022 and by ICVAM in 2024 that outline what criterion an in vitro NAM should meet in order for people to have confidence in using data for that NAM for a regulatory purpose. And you can see there are 5 components: independent review, data transparency and integrity, technical characterization of the assay, human biological relevance, and a definition of the fit for purpose of the assay. And I'll give an overview of this, but if you want a more detailed read, this paper by Sierra Boyd is now available online, and this goes into more detail on how the different assays in the battery map to these different criterion presented by ICVAM and by van der Zalm. And so you'll recall I said earlier that for an assay to be included as part of the battery, it had to meet these 4 criteria. It had to be deemed ready for use in screening and prioritization. There had to be a set of chemicals tested in common across all the assays that were included in the battery. And the data from those tests had to be incorporated into the U.S. Environmental Protection Agency's ToxCast pipeline and analyzed using that pipeline. And finally, a detailed methodological description in a tox temp format needed to be available for the assay, and these tox temps were included in the initial guidance document by OECD. And so on a very high level, you can see that there's already some definition of fit for purpose, of an independent review, of data integrity and transparency, and of technical characterization. So I want to continue to focus on the independent review for another slide. And as shown here in this table from this paper by Sachana et al., all of the assays in the in vitro battery have been published as independent peer-reviewed scientific publications. And so these are the different publications that each of the assays in the battery are describe the initial assay and the initial characterization of that assay. And so again, there's a level of independent peer review here for all of the assays that are in the battery as indicated by their being peer reviewed. Continuing to focus on independent review, I mentioned earlier that a paper by Ball Price, all of the batteries had to be, or all the assays in the battery had to be evaluated according to this method. And in the Ball Price paper, they established 4 different categories of criterion with which to evaluate the assays. For example, biological plausibility, reproducibility, technological description of the assay, transparency of the method, and so on. And then they developed a scoring paradigm for each of these. And phase 1 is method development, phase 2 is performance and reliability, and phase 3 was screening, and graded each of the assays, scored and graded each of the assays according to these criterion. And those scores are summarized here in terms of the overall readiness. And you can see the assays that are in the green box are the ones that were included in the DNT in vitro battery. And so those received at least a B grade for their readiness overall in terms of method development, performance and reliability, and readiness for screening. Finally, with respect to independent review, there has been a 3rd level of independent review, which is the entire battery itself. And so shown here is the timeline for development of the battery and development of the initial guidance document on the battery. And you can see the first draft was ready in July of 2021. And at that point, an expert group from the OECD on developmental neurotoxicity reviewed the guidance document, provided comments that were then used to generate a 2nd draft that in 2022 and early 23 underwent 3 rounds of comments and revisions from the Working Party of National Coordinators of Testing Guidelines from all the member countries of the WNT. And that was approved, the initial guidance was approved in April of 2023. During this time, the Environmental Protection Agency convened a scientific advisory panel of external experts to review the DNT in vitro battery, and the SAP published a summary of the meetings, the minutes from that meeting that provided their comments on the strengths and limitations of the battery. Finally, Saipinian convened a review, and that review was published in this paper by Juberg on the battery, and this was a 3rd independent review by expert scientists. In all 3 of these cases, these were large groups of scientists that were external to the development of the battery who provided comments on the assays and their readiness for use in regulatory purposes. consensus that emerged from these reviews that also goes to the fit for purpose of the assays. And so that's another criterion for establishing confidence. All three reviews agreed that the DNT in vitro battery could be used for screening and prioritization, as well as weight of evidence decision-making types of regulatory decisions. In addition, they all agreed that the battery should be a living process that should evolve as the science evolves. So we've covered independent review and fit for purpose. And I want to go into a little bit more detail on the consensus of all three of these different reviews. So I've taken here in this slide comments actually from the Juberg paper, from the OECD initial guidance document, and from the EPA 2020 Scientific Advisory Panel that directly address their use as a screening tool or their use in terms of weight of evidence types of decision making. And while the Juberg review did not specifically state weight of evidence types of decision making, they stated that for DNT, there are various different approaches, including animal models, epidemiology, clinical studies, and in vitro methods that each have utility, and that integration across all of these assays or all of these methodologies is critical for the accuracy and sensitivity of detecting DNT. And so that is in its essence a weight of evidence type of approach. And again, all three of these reviews agreed that the continued development of this battery needs to go on as the science improves and needs to incorporate the latest and best science. I want to turn now to data integrity and transparency for a few slides. And so that's this component of the framework. While all of this review was taking place, the laboratories that developed the assays were busy testing a lot of different compounds. And the timelines for testing these compounds are shown here. The color indicates for these different sets of compounds where they are in terms of have they been tested and are they available on EPA's computational toxicology dashboard or ToxCast, and the link for that is right here. Some of them are tested and they're pending on the dashboard. Some will be released to the dashboard in the future. These testing sets focused on different priorities. One was testing DNT reference positive and negative chemicals to begin to get an assessment of the sensitivity and specificity of the overall battery. There's also an effort made to test as many chemicals as possible that have been tested in DNT guideline studies so that a comparison could be made between the in vivo guideline study data and the in vitro assay battery data. And then chemicals with specific programmatic interests, such as the perfluorinated compounds, organophosphate pesticides, botanicals, and cannabinoids were also tested in these test sets that are either have been completed or are pending in the case of this EFSA project. So this slide on the left-hand side illustrates the number of chemicals that are tracked by different inventories by the EPA. And on the right-hand side in this histogram, the percentage of those chemicals that we have some sort of toxicity testing data for. As you can see here for developmental neurotoxicity, less than 1% of these chemicals were tested in a guideline DNT study. Overall, there are about 228 chemicals that have been tested in a DNT guideline study in vivo. There are 81 chemicals that have been tested across almost all of the DNT in vitro battery that are currently available in ToxCast. There are another 115 that are pending and should be available in ToxCast by this fall. And another 120 that have been tested are in the process of being pipelined and will be available sometime in late 2025 or early 2026. And then for one assay in the battery, there have been over 500 compounds that have been tested and that are currently available in EPA's ToxCast or computational chemicals dashboard. So these data are easily available, and in contrast to the data for the in vivo DNT guideline studies, it's actually been very challenging to account for all the compounds that have been tested in these studies, and getting the actual data that is presented in these studies can be very difficult to do. And so in terms of data integrity and transparency, comparing the in vitro to the guideline study, the in vitro data has a lot higher transparency. And the nice thing about ToxCast too is you can follow exactly how the data were analyzed, and that's part of the underlying information that's available in ToxCast. Turn to the technical characterization of the assays that are part of the battery. As I mentioned, all of the assays that are in the battery have these tox temp forms, which describe the assays in great detail. And I've highlighted in yellow here the different aspects of technical characterization that are found in the ToxCast or in the tox temp forms. And so there's a whole section describing the general features of the test system source, including where the cells come from, characterizing them, troubleshooting them. There's a section on the test method and the protocols used, acceptance criterion for the test method. In addition, the coefficients of variation, standard deviations, and other metrics of the control wells and z-scores are available for many of these assays as well. So to provide further information, all the tox temp forms are available in Annex B of the OECD 377 document, which is the initial guidance document. And so here's a screenshot of the table of contents, and you can see Annex B are the assay descriptions, and the actual tox temp forms are appendices B2 through B10. And so you can get these from the OECD 377 document. In addition, on EPA's CompTox website in the Exploring Data from ToxCast, you can download OECD 211 forms for all of the assays in ToxCast, including the assays that are part of the DNT in vitro battery. And so this is the link to that site, and you can download then the documents as you see fit for the different assays from the DNT in vitro battery. So in terms of technical characterization, the assays have been well characterized technically, and those technical characterizations are readily available in different places. Finally, I want to turn my attention then to human biological relevance. Here's again the list of assays and their initial publications. 12 of the assays use human cell models, and so would be considered to be directly human relevant in many respects. 3 assays use rat primary cortical cell models, and that's a neurite initiation assay, a neurite maturation and synaptogenesis assay, and the network formation and microelectrode array assay. And so what I'd like to do is spend a little bit of time talking about these assays, because if you read Van der Zalm, in terms of human biological relevance, it said, demonstrate the similarities between the physiology of the test system or the biology measured and human biology. It does not say that the test system must be one that incorporates human cell models. Use the network formation assay as an example, because all 3 of the models that use rodent cells use the primary cortical model. It's the same model. The network formation assay consists of a specialized plate called a microelectrode array plate. It has a 48 wells. Within each well are a series of 16 microelectrodes. Each well is separate from the other wells on the plate. Shown here is a photomicrograph with the microelectrodes, which are about 50 microns in diameter, and then these black lines going away from each microelectrode are the leads that connect to an amplifier. And you can see here there are primary cortical cells being cultured on top of these electrodes, and these electrodes then can pick up spontaneous electrical activity from the primary cortical neurons. And a screenshot of that activity is shown here. You can see there are bursts of action potentials as well as individual action potentials. Each box here represents one electrode from that well, so there are 16 boxes here, one for each electrode. You can see in this particular instance, there's activity occurring nearly simultaneously across all 16 electrodes in this well. And so this is a snapshot in time from each electrode in a single well. And I want to point out that what we're measuring here is the underlying biological activity that is summed in order to give rise to EEG recordings. And so in terms of the relevancy of this particular metric to a human clinical metric that is measured, it's directly relevant. The assay itself, you plate the cells on day in vitro 0. They're allowed 2 hours to attach to the bottom of the plate, and then media is added containing the compound of interest. That media is, and the compound is present throughout the experiment then. Because this is a non-invasive recording approach, we can record on multiple days in vitro. And so typically recordings are done on day in vitro 5, 7, 9, and 12. That's the last day of the assay. After the recordings, media are changed on days 5 and 9. And on day 12, viability is measured using 2 different viability assays. This slide shows how the rat cortical neuron model is representative of what's found in the human cortex. We have both astrocytes and neurons present in this model. They're excitatory and inhibitory neurons, as shown here by both the overall staining and this higher resolution stain of V-GLUT and V-GAT. These cortical cells form active synapses, and you can measure synapses using a marker of presynaptic terminals called synaptophysin, and they have some expression of microglia. In addition, the P450 expression is similar to frontal cortex of rat, but very different from the liver. In terms of receptor types present in functional responses, we know that this primary culture of cortical neurons from rat includes a number of major transmitters and channels that are present in humans, including AMPA, kainate, and NMDA receptors, GABA receptors, nicotinic receptors, and dopaminergic receptors, as well as voltage-gated sodium channels, and not shown are voltage-gated calcium channels. So again, the pharmacology of the model system is representative of what is found in humans. Finally, the activity that we're measuring itself here, the spiking, bursting, and synchronous activity are intrinsic network functions. And so these properties of networks develop spontaneously both in vitro and in vivo. It's well known that neurodevelopmental processes are influenced by electrical activity. And the synchronous activity in these networks is integral to sensory awareness, attention, memory, and other cognitive functions. And lastly, and this is important, the patterns of this network activity are highly conserved. And so there are papers demonstrating that there is greater similarity in the patterns across the same brain regions of different species than there is between different brain regions within the same species. And that's shown here. So here are recordings from MEAs of rat primary cortical neurons, and you can see the intrinsic pattern that arises after a couple of weeks in culture. And you can see how that differs from a rat midbrain culture and from a rat spinal cord culture. And then these 3 recordings are from different human models, human IPS models, that are grown in culture on MEAs. And you can see the pattern of activity here is much more similar among the cortical types of neurons than it is to the rat. And so this is that similarity in activity that is across species in the same region and greater than within the same species in different regions. And for some reason, the eye cell just popped up there at the end. So if we look at all these different criterion for establishing confidence in the use of data from the in vitro battery, We've shown that a number of different independent reviews have taken place, that there's a high degree of data integrity and transparency. These assays are well characterized in terms of technical characterization. The fitness of purpose has been defined for what data can be used for from these assays. And these assays have a high degree of human biological relevance. And so hopefully this establishes some level of confidence in the use of data from these different assays. I'd like to spend the next couple of slides talking about reference compounds for different DNT assays. And shown here is a table from the ICVAM report on the qualification, validation, and quantification and regulatory acceptance of new approach methods using several different terms: positive controls, performance compounds, proficiency compounds. And then shown on the right-hand side here are nomenclatures that were developed in a paper by Kevin Crofton, who's the lead author, that was published in Altex in 2011. And so the terminology that is often used in the DNT field is a little bit different. And this slide will help you map the different terminologies between what is sometimes used as in the DNT field and the terminologies in the ICVAM report. Now I want to talk about DNC reference set compounds, which are compounds where there's a limited amount of confirmed human and/or animal DNT reference positive compounds, as well as negatives. Here is an EFSA scientific report that was developed by Kevin Crofton and Bill Mundy. And it talks about recommended DNT reference set compounds for in vitro assay development. So the purpose of the compounds that are discussed in this document, and this is available online at this DOI, is to help evaluate the ability of a given DNT in vitro NAM to identify chemicals where there is evidence of developmental neurotoxicity in vivo. And so to develop this compound, and they developed reference compounds that are both reference positive compounds as well as reference negative compounds, Crofton and Mundy drew on a number of different sources that are shown here for both the positive compounds as well as the negative compounds. And they categorized them into 5 different groups, and those different groups are shown here. And this represents a curated list that can be organized from more to less certainty of DNT potential. They excluded a number of compounds that had low solubility in water or DMSO, pharmaceutical or chemicals that, such as cocaine, that are regulated, that are not readily available. And so what this represents is, especially in these first 2 categories, chemicals that can be tested in an in vitro assay to demonstrate that it is capable of identifying chemicals that have the potential to cause developmental neurotoxicity. They also mentioned that there are 10 favorable negative reference chemicals, and that those should be tested in order to evaluate the sensitivity and specificity of a given assay and of the overall battery. And so what's coming in the future, and there's a lot going on right now that we don't have time to go into in this series. There's a lot of work going on to improve the assays as well as our biological understanding of the assays. And so some of the assays are being multiplexed and scaled up and moved to more current imaging platforms. That's going on with the EPA's proliferation and apoptosis assays. There are 2 papers that should be out sometime in the next year looking at the role of important neurodevelopmental and disease pathways, such as Notch and EGFR, and shown here. And these are the EPA assays, and Ellen Fritsche's lab is doing a similar exercise with her assays. And so this will provide a greater biological context for the different pathways that are captured by each battery. There's tiered testing approaches that are being worked on. So a tier 1 assay that's being worked on in Josh Harrell's lab is high throughput phenotypic profiling. My laboratory has been working on a brain sphere model on high density microelectrode arrays, which we hope to have published in the near future. And then there's work on the next generation of DNT NAMs. And you recall that I mentioned earlier that all of the large groups that reviewed the DNT in vitro battery said this should continue to evolve as the science evolves. And so we're looking at next generation approaches that might include measures of plasticity, increase the biological relevance, and a larger characterization of the biological applicability domain. I'm going to go into assay transfer to naive laboratories in just a moment. There's also work going on comparing different data evaluation pipelines. So I mentioned the EPA pipeline, but there are also some other pipelines where the data are made available. And finally, there's work going on QIV-IV modeling for the neurosphere assay. And so there are a lot of things going on now. And so these assays will continue to evolve as the science evolves. And I think there will be some really nice things coming out in the future. So in terms of assay transfer activity, I want to focus first on the microelectrode array network formation assay. EPA has been working with Cyprotex to transfer its network formation assay to Cyprotex, and we have data back now from Cyprotex and are in the process of analyzing that data. We've also established CRADAs with both the IUF and DNTox to transfer all of the EPA assays and are working with both those groups, and testing is currently underway with transfer of these assays to DNTox and IUF. And then we're also working with Cyprotex to transfer our human neurite outgrowth assay, and we hope to have data on 200 compounds in that assay by this fall. And finally, we've been working with FDA and collaborating with them and Megan Culbreth at the FDA, and Megan has tested 6 performance compounds in the network formation assay, 11 compounds in the proliferation apoptosis assay, and 16 compounds in the human neurite outgrowth assay. And so analysis of that data is all ongoing and should be out this fall sometime. Excuse me. And so this is one of the major efforts that's being made now, is to transfer these assays to other laboratories, because in terms of establishing confidence, this is perhaps one of the areas where much more could be done, and this will help to address this in terms of establishing confidence, in terms of replicability from one laboratory to another. And so then to conclude, there's been this DNT in vitro battery that's been developed on understanding chemical perturbations in key neurodevelopmental processes, and disruption of those processes is associated with adverse neurodevelopmental outcomes, and chemicals known to cause developmental neurotoxicity can disrupt these key neurodevelopmental processes. And these can be mapped to adverse outcomes that are evaluated in the current guideline DNT studies. There have been 3 comprehensive independent peer reviews of the battery that find that it's ready for fit-for-purpose screening and weight of evidence decision making. When we evaluate the battery against the ICVAM criterion for confidence in the use of DNT NAMs data, it shows that the battery covers multiple elements of all 5 components needed to establish confidence. And so assay transfer activities are ongoing and showing promise, and that the evolution of the battery and the assays will continue as the science improves and moves forward.
Hello, welcome to this webinar on application of the DNT in vitro battery within integrated approaches to testing and assessment, or the so-called IATAs. My name is Mangas. I'm a regulatory toxicologist, and I work since 2018 in the pesticides peer review unit of the European Food Safety Authority, and represent EFSA in the DNT in vitro battery expert group of OECD since then. In this short presentation, the main goal is to promote a better understanding of the key concepts of the IATA, or integrated approaches to testing and assessment, framework for developmental neurotoxicity and the different elements for the weight and integration of the evidence of the DNT in vitro battery into regulatory decisions. This is an introductory presentation and is based on the case studies that use the data from the DNT in vitro battery and to June 2025. There will be detailed presentations on different examples of case studies with different problem formulations and on how the DNT in vitro battery data has been interpreted for different contexts of use, and this will be available in session 2. The history of the integrated approaches to testing and assessment for DNT, in line with the history of the development of the DNT in vitro battery, is an history of a collaborative effort. When in 2016 OECD and EFSA organized the DNT workshop, and there it was agreed that the DNT in vitro battery, the assays that were assessed as ready by the different stakeholders, were ready for screening and prioritization, but that further work was needed for using them for hazard identification and characterization. Among this work, in addition to the testing of chemicals and other activities that were already presented by my colleague, the development of IATA case studies for the regulatory implementation was also noted as key. So more or less in parallel, the two activities, the activity of the development of the OECD DNT in vitro battery guidance that so far has been approved by WNT as an initial recommendation document in 2023, and the development of several IATA case studies for regulatory implementation under the OECD IATA program, but also under different programs by the different regulatory bodies, has been working in parallel. I have to say that there are many also IATAs ongoing, and at least for several stakeholders, among them EFSA, there is an ongoing program of developing new case studies that will be running at least until 2027. So this presentation must be seen as a live document that will be changing in the next years. So I invite you to go to the OECD initial recommendation document, Appendix D, and see there the different IATA case studies, and also to go to the OECD website for the IATA case study program, where you have the different IATAs for developmental neurotoxicity. When you do so, I thought it would be important to have in mind some relevant definitions for understanding the concepts. Among them, the concept of test battery. And I have to say that there is no OECD definition for test battery, and there are various definitions more or less in line. So a test battery is a series of tests independent of each other, generally designed to complement each other and to measure different components of a multifactorial toxic effect, and which are usually performed at the same time or in close sequence. Test battery normally tend to complement each other, but they are not integrated into a strategy, as is the case of the DNT in vitro battery. So we are talking about a group of assays that are conducted together for a specific purpose, and that provides you a prediction for a specific toxicity endpoint. In this case, developmental neurotoxicity. The results of each individual assay could be equally weighted, as is the case of the DNT in vitro battery so far, or it could be development of a statistical weight for the different assays to better model the in vivo response. When we are talking about tiered testing strategy, it has to be considered that this is about a sequential assessment. So the result of one tier is used to determine the next step, and a decision tree is needed. so where the uncertainty is decreasing with the years. A defined approach, according to OECD, consists of a fixed data interpretation procedure that can be with a statistical or mathematical model that is applied to data. Can be to different in silico, in chemical, or in vitro data, as could be the DNT in vitro battery, generated with a defined set of information source to derive the prediction. So the defined approach is rule-based and is very rule the data that is included and how the data is analyzed. There is no room for expert judgment. When we are talking about data interpretation procedures, we are talking about fixed algorithms, mathematical algorithms for assessing, for interpreting the data of several information sources, as could be the DNT in vitro battery. The output of the data interpretation procedure is the prediction of the biological effect of interest. The data interpretation procedures are rule-based, and also there is no room for expert judgment here. When we are talking about integrated approaches to testing and assessment, this is a flexible approach. It's based on the integration of multiple sources of information and weights that assess all the existing evidence or all the relevant existing evidence and the main relevant point is that the result can be the generation of new data when required to inform regulatory decision making regarding potential hazard or risk. And IATA necessarily includes a degree of expert judgment in the sense which information source you are going to include, which method you are going to use for the weight of evidence, which uncertainties are you able to accept depending on your problem formulation. So there is this flexibility. However, part of the IATA can be defined approach to testing and assessment and can be more or less standardized. In an IATA, and you will hear this a lot, there are different information sources that can be included or also in the defined approach. Here we are talking of the different lines of evidence that you consider in the weight of evidence. Can be physicochemical properties, testing methods, non-testing methods, or any other source of relevant information that you consider in your weight of evidence. Then you will hear a lot about AOP informed IATA and the use of AOPs in the IATAs for developmental neurotoxicity. There are two ways that an AOP can be used in an IATA. The one I have called AOP informed IATA, and you will see various examples in the EFSA IATA case studies where AOPs are used to inform specific parts of the IATAs and to provide a structured way of organizing all the information that is included in the weight of evidence of the IATA. Allows you to identify data gaps on the knowledge, on the biological knowledge, and to make regulatory decisions, even to develop testing strategies for the gaps. But you do not need to have an AOP endorsed by OECD to develop an AOP-informed IATA. An AOP-based IATA, the AOP serves as primary framework for the development of the IATA. So your starting point is an AOP endorsed. You develop test methods for the different key events and for some of them, and then this is the basis of your IATA. This is your only lines of evidence for the IATA. You can see examples in the skin sensitization, for example. Then another topic which is very relevant and you will hear a lot in the IATA case studies example is the standard IATA workflows. The standard IATA workflow or IATA framework template allows you to to organize testing and assessment tools, and to propose suite of methods to address unspecific questions or different questions and increase the weight of evidence. So it's a way of moving to a standardization of an IATA for a specific endpoint. Under the OECD umbrella, starting more or less in 2018, several stakeholders, as you can see here, not only EFSA, also US EPA and the National Institute of Health and the NTP program in US, but also UTOX-RES or BIAX, started to develop case studies integrating the DNT in vitro battery for different regulatory purposes, as you will see. And the reason of doing so is, this is part of the regulatory relevance of the DNT in vitro battery, is because there is a need of improvement of the efficiency and in the safety decisions when we assess developmental neurotoxicity. There is a recognition that we need a faster and more relevant approaches across the different chemical sectors when we assess developmental neurotoxicity, and that we need having more information on developmental neurotoxicity potential hazards for thousands of chemicals. And we need a better understanding or a better implementation of the DNT in vitro mechanism and in the regulatory decisions, in our current regulatory decisions on developmental neurotoxicity. However, we need to take into account that the purpose of the OECD initial recommendation document is to provide guidance on how to evaluate the data of the in vitro assays. So what means a hit versus non-hit, what are the uncertainties, what is the biological coverage, what the assays are measuring, and everything that has been explained by my previous colleagues. It is not intended to guide the results of the use for human health risk assessment. And this is because a specific criteria for SCAHT should be developed by the different regulatory agencies depending on the regulatory frameworks and depending on the context of use, and are the different regulatory agencies in these regulatory frameworks who should determine the acceptability based on their needs. It's true that the document contains a list of regulatory needs that could be addressed by the DNT in vitro battery, and the case studies provided there are examples on how this can be done. However, we need to take into account that the different chemicals regulations can vary and vary in the data requirements, and not only that, on how this data, on the flexibility to fulfill these requirements. That is the reason that most of the samples are in the pesticides area. But we need to consider that this is also true when we are taking into account in our regulatory decisions test guideline data, OECD test guideline data that is binded to the mutual acceptance of data. The mutual acceptance of data is sharing only with member states that have the same data requirement. So you will see in the samples the target chemicals and target use of the DNT in vitro battery, and this is from the OECD initial recommendation documents, and the target chemicals are industrial chemicals, pesticides, environmental contaminants, including metabolites or environmental degradants. Of course, there are the limitations intrinsic to testing in vitro that depend on the physicochemical characteristics of the substances in terms of volatility, high reactivity, hazard level, and limited solubility in the media of the assays and on the MSO. This is clearly described in the ToxTem assays in Appendix B of the document. As regard of target uses, it can be divided, and the case studies are divided in two main problem formulations. the screening of large number of chemicals, as we can have TOX21 or Health Canada prioritization of SCAHT, or single chemical hazard assessment, hazard identification, hazard characterization, or risk assessment as part of a weight of evidence-based IATA. And these are the current target use and target chemicals for the DNT in vitro battery use, as you will see in the case studies. It is important when assessing the case studies and when studying them to have in mind clearly three different concepts. The first one is the key neurodevelopmental processes that has been very well explained by my previous colleagues, but just to note that we are talking here of assays that measure early or cellular biomarkers of effects. They are not based on molecular initiated events as the estrogen receptor battery, since this is impossible now to develop a battery on molecular initiated events for developmental neurotoxicity. The assays that are included are those that were deemed ready using readiness criteria that is in line with the current ongoing discussion for the guidance 34 readiness criteria. When we talk about weight of evidence, there are several guidance. I think each regulatory body has its guidance of weight of evidence. There is the OECD 311, EFSA has its guidance, but at the end, the weight of evidence approach is strong, and it is a process more or less systematic that allows to evaluate the strength and the quality of the evidence from multiple sources. And it indicates you how to ascend, weight, and integrate the evidence to reach a conclusion. So here you consider reliability, relevance, and consistency. And you will see different ways of doing this weight of evidence in the case studies. All of them have been followed of EFSA guidance or OECD guidance or other guidance, as can be the USPA ones. And then when we talk about IATA, it's important to remind that the IATA allows you to balance the uncertainty in the data with the uncertainty that is acceptable for the regulatory decision. So the series of case studies are proof of concept case studies for this different balance on the uncertainty on the data and the regulatory decision. So why IATAs for developmental neurotoxicity? The short answer to that is to move towards a better testing paradigm for chemicals for developmental neurotoxicity. This is the current tool that allows you to integrate NAMs to solve complex endpoints, but also provides flexible solutions for a variety of regulatory contexts, as we are having here, and a variety of regulatory frameworks. But importantly, also allows you to integrate the data with the different sources of evidence and to combine the different tests. So it's the appropriate tool to integrate the DNT in vitro battery so far with all the available information that can have a dossier of a chemical and establishing the weight of evidence, and allows you also to use evidence-based systematic approaches. I have to say that also allows regulators to become familiar with the different technical elements of the methods and assess the reliability and the relevance, and is a very helpful tool to integrate innovative approach and to decide at some point which one of them are suitable for becoming OECD test guidelines or defined approach. As I said before, it's important to take into consideration that the IATAs are not bound to mutual acceptance of guidance. The IATA concept is not new. Already in 1999, by the Integrated Testing Strategy ECBAN Task Force, the IATA concept was introduced. So far, in the OECD IATA case study project, there are 37 case studies. From them, 5 are for developmental neurotoxicity. And there are also a series of consideration documents and lessons learned of the OECD that I also invite you to go to the 2022 lessons learned, because it's where the DNT in vitro battery case studies were discussed, and some relevant points can be found there. So this is the IATA, the general IATA framework, the general AOP inform IATA from the OECD 2016 document, and everything starts with a problem formulation and gather the existing or the relevant information considered. Then the next step is the weight of evidence assessment, and importantly, an IATA allows you to decide whether you have sufficient information to conclude or whether you need to generate additional data. And this can be done in the different steps of the IATA. So again, you can do the weight of evidence and decide whether you have sufficient evidence to conclude on your problem formulation, or you need to generate new data. As I say, AOPs can be used to inform the IATA and can also be used as a framework to develop the IATA. When we are talking about the DNT in vitro battery and AOPs, I took this figure from the OECD initial recommendation document. We need to consider that we are talking about key neurodevelopmental processes, so biomarkers of effects, of cellular response, something early or medium effects in an AOP framework. So the AOPs are used and can be used to develop the Assays and also to interpret the Assays. However, we need to consider that the number of AOP endorsed for developmental neurotoxicity is limited. This is because of the complexity of the developmental processes and physiological feature of the developing nervous system, as you have here. This makes it challenging to map the neurodevelopmental toxicity pathways and to link them to a specific adverse outcomes and to a specific critical windows of vulnerability. But this is also because to develop an OECD endorsed AOP is very demanding, and sometimes there are no other resources available to do that work. Of course, the development of DNT OECD endorsed AOP is very much encouraged by the DNT in vitro battery expert group and by EFSA. So this is how the DNT AOP landscape looks like now. There are 11 DNT AOPs in the wiki. Here you have the numbers in the case you are interested. From them, 5 are OECD endorsed, including this one that was developed by EFSA in the last year and recently endorsed. Binding to voltage gain sodium channel during development leads to cognitive impairments, and that maps here the neuronal network formation assay of the DNT in vitro battery. There is a lot of work ongoing by many stakeholders, so I am sure that in the next years this landscape will dramatically change. But so far, all of them are qualitative. They are very helpful to understand the DNT mechanisms. They are very helpful to correlate with the human neurodevelopmental disease, and I invite you to read the AOP 442, where you have a very thorough systematic analysis of the literature for this specific pathway, and also a very thorough analysis on the uncertainties. And of course, if one day there is a full network of AOPs or an AOP network for developmental neurotoxicity or various networks, this will allow to develop computational models. However, so far and in the next years, an approach based on AOPs, so assays that are anchored to endorsed OECD AOPs, key events, is a limitation, because we will be able only to detect very small number of positive hits, because very limited key neurodevelopmental processes are so far mapped into OECD endorsed AOPs for DNT. I would like to give a little bit of details on the text that you are gonna find in the OECD initial recommendation document on the elements for establishing the weight of evidence within the IATA. This is the longest section of the OECD document. I really invite you to read the details there, where you will find the generic in vitro uncertainties or weight of evidence issues. We need to consider that the DNT in vitro battery data so far adhere to the OECD guidance 286 and the OECD guidance 211, but is a non-test guideline data, and this must be considered in the reliability and relevance assessment of your weight of evidence. In addition, there are a number of specific limitations or uncertainties of the DNT in vitro battery, as can be the unknown metabolic competence, or the limitation of the testing, as I already said, for some of the chemicals as volatiles or non-soluble. It is key, and a lot of work has been done, and we need to thank our colleagues from USCPA for including all the raw data in SCAHT and having transparency in data sharing and data analysis. There is a section on individual assay interpretation and also another on evaluation of the data from the entire battery of assays, in line with the presentation of my previous colleagues. In short, what this weight of evidence section is telling us, regulators, is that you need to treat the data of the DNT in vitro battery as you treat the other data for your weight of evidence in hazard assessment. In this section, you will also find a summary of the current limitation of the evaluation of the data, and also as a result of the IATA case studies. And one of the main ones is that there is no, so far, but hopefully we will have soon, a consensus set of DNT reference chemicals for use in development and further validation of the DNT in vitro battery assays. This we should not overstate, because it will allow estimation of the performance in terms of sensitivity and specificity of both the individual assays and also the full battery. There is the need of development of additional AOPs, but we have already talked a lot about that. And we can say that there are still relatively limited number of chemicals tested. And there is a need of a consensus-driven IATA standard framework, workflow, or decision tree. So, in a way, the DNT IATA case study, this international collaboration program, has been created to create evidence to move towards acceptance for different contexts of uses. And it is allowing us to develop standards for evaluating the suitability of the DNT in vitro battery, considering the current approaches and considering the current validation status, but also to identify aspects that can be standardized. And in terms, for example, the reporting of the weight of evidence or the evaluation, data interpretation procedures, and so on, is allowing us to define more specifically the context of uses, also understanding their different regulatory needs for a variety of regulatory contexts. And in a close future, it will allow us to select which assays are used for developing test guidelines, or will allow us to develop defined approach or defined approaches for developmental neurotoxicity. So the case studies and the work done by several stakeholders have a further purpose of evaluation of the reliability of the relevance and mapping the uncertainties, but also gaining confidence in extracting, interpreting, and using the data of the DNT in vitro battery, understanding the data of the battery as an AM. I am confident all this work in the IATA case studies will help to develop a decision framework in a short timeline, as I say, that can be a defined approach or tier testing strategy, and to define more specific criteria for the different regulations and the different policies to incorporate the data into regulations. So in the samples, you will see that there are several contexts of uses where the DNT in vitro battery has been implemented. You will have examples of solutions for a variety of regulatory contexts, different problem formulations. We have data-rich, data-poor chemicals. We have examples across different chemical sectors, not for all, across different regulatory frameworks, and importantly, for various problems formulation. Of course, further development of case studies is very much encouraged by the DNT in vitro battery expert group and also by EFSA. So the case studies can be organized in those for screening and prioritization, hazard identification, hazard characterization, and risk assessment or point of departure derivation. Of course, when you go to point of departure, you need to have more confidence in the data and less uncertainty. You can find here in this table a comprehensive list of the IATA case studies for hazard identification and characterization, including point of departure derivation, that integrates and uses the DNT in vitro battery up to June 2025. I have to say that all the samples are from pesticides risk assessment. You have here the reference where you can find further details, and you will see several of these samples in the session 2. But it's true that we have different case studies from different stakeholders developed as EFSA, OECD, or EU Tox or industry. In the second table, you have the samples for screening and prioritization problem formulation, and we have 3 main samples: screening on PFAS, ongoing industry case studies on screening and prioritization, and also the organophosphates flame retardants samples that you can find in the different references included here. So to conclude, as you can see, and as you will see more details in session 2, the DNT in vitro battery is being used by different stakeholders and by different regulatory bodies within IATA case studies for different problem formulations. We can divide so far the case studies in the ones for hazard identification and characterization and the ones for screening and prioritization. So far, the DNT IATAs has been trusted by stakeholders from industry to risk managers for some case studies and allowed the acceptance of the DNT in vitro battery as non-data also from industry to risk managers. The IATA has been demonstrated to be a relevant tool to include the DNT in vitro battery mechanistic data in the risk assessment, as you have seen in all the samples. For hazard identification and characterization, the most of the case studies, all of them use pesticides as target chemicals. However, similar weight of evidence approaches has been done by different stakeholders. Other problem formulations are possible, and it is encouraged to develop further IATA case studies as could be for classification of chemicals. And a standard IATA workflow will allow reproducibility of the IATA conclusions for same problem formulations, and we have a very good example with the acetamiprid case study that was first developed by UTOX and then also in parallel by EFSA, where similar conclusions were drawn by two different bodies. Transparent report and completeness of the documentation is needed in IATA, and again, the lack of existing AOP is a limitation, is an uncertainty, but the knowledge of the critical processes is well established, as you will see in the initial recommendation document. So what's coming in the close future in terms of IATA for DNT? There is an DNT-IATA framework template and further interpretative guidance ongoing in a project led by OECD, US EPA and EFSA under the Working Party for Hazard Assessment, and this will allow to leverage on the available DNT-IATA case studies that, as you have seen, are a lot. It will allow to develop additional case studies for some problem formulations where we do not have case studies, and it will use also the new IATA OECD framework developed or under development this year. That is an evolution of the format for reporting the finite approach, and it will allow to standardize how we develop the IATAs for DNT. So this will allow to, in a faster way, apply an existing IATA for a new chemical or modifying the existing IATA to add new data. The idea is to apply the IATA framework for different regulatory frameworks and different problem formulations for different contexts of uses. So it will be a flexible framework, but that will have some of the parts more standardized, and it will include the data modules of DNT that can be included or excluded under rationale. As part of this DNT-IATA framework work, it will be specified which part can be standardized with data interpretation procedures and which parts still needs more flexibility to be maintained. This will allow, of course, further standardization of the uncertainty analysis for DNT in general, but also in particular for the DNT in vitro battery. As part of the framework, there will be a specific guidance for PBK modeling and EBIF extrapolation for developmental neurotoxicity. That a document has been already proposed to the WPHA. And that will be integrated in the IATA framework with specific considerations of placenta and blood brain. Further standardization of the data analysis of the DNT in vitro battery is also ongoing, and this has already been presented. And the results of the ongoing testing of chemicals and the transferability data of the Assays of the DNT in vitro battery will be also ready in the next years. A consensus DNT reference chemical list will be also available in a very close future, and all of this will be a very huge step towards having a defined approach or defined approaches for developmental neurotoxicity assessment of chemicals. With this, I thank you very much for your attention and for listening, and also for your interest in the DNT in vitro battery. But I have to thank also to the big DNT-IATA teams, to all the DNT-IATA developers that, as you have seen, are a lot, collaborators and reviewers, to the OECD DNT in vitro battery expert group, the OECD-IATA case study project, the Working Party for Hazard Assessment, and WNT members, the EFSA-IATA Neurotoxicity Working Group members that developed the EFSA-IATA case studies, and the EFSA Toxicology of the Pesticides Peer Review Unit. If you have further comments or questions on the IATA case studies, please don't hesitate to write me to my email address that is there. Thank you very much.